Dr. Peter Jones is the Mick Hitchcock Endowed Chair of Medical Biochemistry at the University of Nevada, Reno. He's been working on mechanisms of disease in FSHD since 2003 with his wife and co-PI Takako Jones. He works on epigenetic mechanisms of gene regulation, such as DNA methylation, post-translational modifications, and chromatin regulators. Some of those we've heard about earlier. The goal is to develop therapies that target these novel mechanisms, taking advantage of approaches such as CRISPR and other small molecule designs. So we're very fortunate to have him as a West Coast colleague, new as of January this year, and to have him here with us today. Thank you.
I'm going to time myself because those who have seen me before know I will talk extremely fast. First thing, I like to make sure that it's very clear we are a husband and wife team. We're co-PIs. We've been working together day and night for every day for 10 years. We're still standing and doing okay. She describes the Japanese view of it as I'm the head and she's the neck, but I always have to give the seminars, but she will answer all of your questions. So I'll also be a bit more sciencey than what you've had. We are PhDs. We don't see patients. We're not MDs. We are very bench-oriented, and we don't get out of the lab much. I will say, first I have just brief disclosure. I do work with some pharmaceutical companies, and that should be good news. We need pharma if we're ever going to get things to clinic. So I will talk very fast because I get very excited. We love what we do, and it's very exciting times. The take-home message of my talk is that from the outside, it may not look like a lot's going on. From the inside, this is an extremely exciting time in FSHD research. There's a tremendous amount of activity in respect to therapeutics going on behind the scenes, and the future, near future, is really bright, in my opinion, for FSHD. And the fact that pharma companies are actually interested in FSHD, and where they weren't 5 years ago, is fantastic news. So sometimes it might feel like you're a little bit alone. You're in a rare disease. You may not know other people with FSH. So, really, neuromuscular diseases are actually quite a large class of diseases. There's at least 9 classes of muscular dystrophies, of which FSHD is one of. And the reason I bring this up is because I get emails from patients all the time, and you're free to email me. But normally they hear in the news about something in Duchenne or some other disease, other muscular dystrophy, and say, what does this mean for FSHD? And the general answer is it's good news, because the way we approach many of these diseases, the technology that's being developed for these other diseases is often applicable for FSH, and what we do in FSH is often applicable beyond. So it is actually a big worldwide effort that will be beneficial to FSHD patients. Aging, everyone here, almost everyone here, has a disease of muscle. If you're over 35, that's about what I figured when I started really aging. You're basically losing some muscle mass. And some of the research that's involved in aging, some of what we learn in muscular dystrophies and what we learn in aging are cross-applicable. Okay, so this is actually a bigger area of research than FSHD alone. In fact, the way MDA even puts this out is, they really make the point that scientific and medical breakthroughs across the neuromuscular disease accelerate treatments and cures in all the neuromuscular diseases. Okay, so this is actually, it's very important. What we learn from one, we take into the other. All right, so I mentioned that I got into this in 2003. I am not a muscle biologist. I had never heard of FSHD until this kid came into my office, and he had been to one of these meetings in Baltimore in 2002, and he had been diagnosed with FSHD, and he had heard that maybe FSHD was epigenetic. He saw on my website that I work on epigenetics. At the time, I worked on a disease called Rett syndrome. And he came and he told me about FSHD. I lied and told him I knew what he was talking about. I had to find the FSH Society website to figure out what he was actually talking about. And then it turned out that, wow, this might be something that I could use my expertise on. And then he ended up getting his PhD in my lab, and now we're a full-time FSHD lab, and that's pretty much all we work on. But the key to it, and from our perspective, is this idea of epigenetics. And this kind of builds upon what Dr. Day talked about earlier. The only other thing you really need, I'd like you to take home from this, is what does the term epigenetics mean? Because you'll hear this a lot in the news. You hear this a lot. So Thomas Hunt Morgan, my favorite Nobel laureate, described it as treasure your exceptions. This is genetics that doesn't work like genetics is supposed to work. Okay, non-Mendelian. You basically, if I had a pointer, I'd show you that calico cat. The reason it has that color is because the gene that causes the color is on the X chromosome. That is sporadically X inactivated because they're all female. That's why all calico cats are female. And the context of the gene affects the gene as much as the sequence. Okay, so it's context-dependent, sequence-independent gene regulation. And this can be influenced by the environment, things like diet, aging, etc. And I think the most spectacular example of this is these mice. So these mice, these are genetically identical mice. We can do this in the lab. They are identical at all locus. They're epigenetically different. Their mothers were fed different diets when they were in utero. And what happens is you can come out as this brownish color, you can come out as this yellowish color. They are genetically the same, they're epigenetically different. The context of the agouti gene is different from between these mice based on the developmental environment. Okay, so who cares if you're yellow or you're brown? Well, the mice care because these yellow mice are going to be obese and they're going to spontaneously develop tumors compared to their genetically identical agouti mice. Okay, so epigenetic effects, environmental effects on your genome. The DNA sequencing was easy. The epigenome is what actually, in my opinion, is very important. This affects long-term health, it's heritable, but the key is it's manipulatable. You can manipulate the epigenome without doing gene therapy. You don't have to change the gene sequence. What that means is if you have an epigenetic aspect to your disease, it's amenable to small molecule therapies. Essentially, we can shift the epigenome without affecting your gene sequence. And so this is great news. And it turns out there's a lot of epigenetic diseases. I could talk for an hour about it, but I won't. But here's FSHD. Okay, so FSHD, the reason we're interested in working on it initially was because it's an epigenetic disease. We saw that there's these deletions, but actually the green and the yellow indicates that the FSHD region in FSHD patients is a different environment than the same region in healthy patients. Okay, and in FSHD2 it's the same thing. Now there's this really, really funky thing that goes on with FSHD. About 1 out of 6 patients, 1 out of 6 healthy, controlled, non-FSHD patient from a family that came into the clinic at Hopkins turned out that they were genetically identical to their relative who has FSHD, but they were healthy, they were fine. Turns out they were epigenetically healthy, even though they were genetically FSHD. Nature has cured this, okay? You don't need to go from FSHD to healthy. You need to go from FSHD to asymptomatic, right? This means we can actually attract this. And this is one of the reasons pharma is very interested. This is something you can do with small molecules. Nature has cured it. We just got to be as smart as nature. So, 2003, I got into FSHD, very small field, okay? Gene, we didn't know. Pathogenic mechanism, didn't know. Models, they were wrong. Animal models, didn't exist. Treatments, they said, oh, what do you do for DMD? We'll try it for FSHD. Not a dumb thing, not bad. What else are you going to do? We didn't know anything. Thanks in part to my student and the field now, more than 70 labs worldwide. Flash forward ahead, we know the gene. Now you know the gene, you have a target. Okay, if you don't have a target, what are you going to do? We have a target. Mechanism, epigenetic dysregulation. A lot of other targets I'll show you. Cell models, now that we know what we're doing, and with help of patients that have donated biopsies and cells and family members and healthy family members, we have a lot of cell models. Now we got tools. Animal models, we have mouse models, frog models, mice models. It may sound stupid, but you know what? Investors, pharma, they like to see a mouse that's not walking around. You give them your drug, and now it's walking around. Okay? That's powerful. Okay, FDA actually doesn't care so much, but because a lot of mice get cured, and that doesn't necessarily translate. All right, so this is the simplified model of what we know now. And the good news about this, DUX4 is the key player, as Dr. Day pointed out. Everything upstream and everything downstream, these are all potential therapeutic targets. These are points of intervention. You don't want one point of intervention because what if you can't do it, right? So we have actually numerous points of intervention that we can go after. So DUX4 directly. There you can actually get antisense molecules, morpholinos, specialized microRNAs. Basically the idea, can we destroy the DUX4 mRNA? I think that's enough. That's only one mechanism. There are many pharmaceutical companies and research labs using this approach, but that's only one. How about upstream? Let's say, as Dr. Day pointed out, the problem is DUX4 gets turned on aberrantly. Okay, let's not let it get turned on. Can we get a drug that basically works upstream of DUX4 so that it never gets turned on? Or maybe not that it has to be turned off, but let's just dial it down. In FSHD, if it's at 8, let's dial it down to 4, to the asymptomatic level. We don't need to go on to off. We just need to take someone with symptoms and dial them back to an asymptomatic case. Protein, downstream, immune response. We know you get a great immune response. Part of the problem is your immune response is giving you problems. And then there's a number of other factors downstream that can actually be involved. Bottom line, there are a lot of points of intervention, a lot of really smart people out there working on all these points, and a lot of pharmaceutical companies that are interested as well. Small molecule inhibitors, let's block the effect of DUX4. Okay, good news. Finally, you didn't hear about the myostatin. DUX4, Acceleron, you got a clinical trial. And actually, I could mention previously the ATIRE trial that some of you may be aware of, right? That's functioning right down here. It's saying, let's block this and see if we can block the effect. Okay, the Acceleron trial is this, again, independent of DUX4. You're still making DUX4. You're still got your FSHD muscle, but let's just make more of it, and hopefully you're bailing the boat out faster than the boat's filling up. We'll see how that works out. All right, so this is what we work on. You can't work on everything. We're a small group. We do small molecule epigenetic effectors. Makes sense. We're an epigenetics lab. So we're trying to see, can we return the pathogenic or the FSHD state of the region to an asymptomatic state with small molecules. And then also we're doing it artificially with this thing called CRISPR that you may have heard of, unless you've been living under a rock. And we'll talk about that in a bit. Okay. I get a little excited. All right, so the question is, can we therapeutically dial down DUX4 expression? If we look at DUX4, see, this stuff is presented in black and white. People love black and white. Let's see, on and off. The reality is, someone with FSHD is expressing a lot of DUX4. Someone that's healthy is actually expressing a little bit, just not on and off. And someone that's asymptomatic, well, you have a mid-level. So again, we don't have to turn this thing, we just got to dial it back. This is part of actually why there's so much variability in patients and symptomology. You have different levels. You're epigenetically different, even if you're genetically very similar. And so we just got to dial this back. Can we do that? So this is some of what we've learned in the past 15 years. All you need to know from the cartoon standpoint, all of these factors keep DUX4 off in the healthy, and when it's screwed up, these factors are involved in keeping it on. So what do you got to do? You got to block one of these factors and maybe even prevented it from being turned on. Okay? So everything over there is a candidate for small inhibitory molecules. We're going to go through and say, what happens if we knock one of them out? And can we take FSHD and take it from symptomatic to asymptomatic? Can we dial it back, dial back the badness? And the answer is, just giving an example in the lab, yes, we can. We know enough about the basic biology. This is basic science. How does this disease mechanism work? How is this gene turned on? Dead boring, right? It's boring until you find out that if I take one of those factors out with a small molecule, DUX4 goes all the way down. Your muscle genes that make you a muscle stay perfect. And all of a sudden, you got a great candidate for drug therapy. Okay? All right, so targeted repression of key epigenetic regulators reduce pathogenic DUX4 expression. Okay, we can do this in theory, and actually there are companies that are working on doing this because we want to get a small molecule that will actually do this. So, FSHD, the second thing we do is called CRISPR. So now I just showed you we want to kind of naturally revert it, but there's new technology out there where you can target the genome and manipulate the human genome or the plant genome or whatever genome you're interested in. And so can we artificially do it? If we can't get a drug, maybe we can artificially shut off the region. Okay, genome editing. Everyone's scared to death, right? We're making super babies. Well, we're not. That's not the whole story. What you need to know about CRISPR, all CRISPR really is, if you've heard of it, is a genome targeting. okay? It is a mechanism stolen from bacteria. Again, basic science, basic research that produces huge results. Sequence-specific genome targeting. I want to take this factor to this part of the genome. That's all CRISPR is, right? Sometimes you're cutting it, sometimes you're doing things. What we want to do is we want to take something to the FSHD region and shut it off, okay? So instead of, if we can't find a drug, we'll just bring a big off signal that says, go back to being off. Okay, so actually at UMass developed a system, a dead Cas. What this means, it does not cut the genome. It has a repressor domain. This is a function of a protein, someone from another protein that turns genes off. And we showed that we can target the FSHD region and shut off the DUX4 gene in human cell culture. This may not sound like a big deal, but actually, turns out, we're the first people to do it. And it is a big deal because everyone else in the world is cutting the genome, it appears. FSHD, we don't need to cut it. We don't actually need gene therapy. We don't need to remove anything. We just need to shut it off. Taking a piece of the genome that wants to be off and help it, okay? Off-target effects, not that big a deal. FDA point of view, if you're doing genome editing and cutting the genome and it doesn't turn out so good, you're out of luck. You can't fix it. We can remove this drug. So it turns out everyone got very excited. Then we realized, whoa, this was actually a side project. And it took us 4 months to do this. But all of us, we showed that actually you can use this cutting, FSHD, we are riding on the shoulders of brilliant people in other fields and just co-opting that technology for FSHD, putting us up, I think, at the cutting edge of at the research. Advances in biomedical research help all. We got to keep the funding going. So can this actually become a therapy? That's the question. Well, there's a number of companies that think, yes, it can. In fact, it's estimated to be a $5.5 billion industry. If there's money to be made, someone's going to figure out how to make it. That's, again, good news, right? So these companies are private held. The patents, kind of funky. The bottom line is that there has been a CRISPR trial getting the green light. This is cutting. Ours is safer than this cutting. Ours is even better. In fact, if I had a CRISPR company, I couldn't think of a better disease to work on. Now, the problem is, how are you going to get this to all of your muscles, right? Because right now, everyone doing CRISPR does easy stuff like liver and blood. You can get stuff there, just everything goes there. But this is tough, right? Well, we don't have any animal models. Well, we didn't until my genius wife developed an animal model for FSHD. And without getting into it, because I could give you an hour talk on it, here's a mouse that does not have FSHD. Okay, this is our simple assay. You talk about assay tests. We can't hook them up to an Xbox, but we can just tip them over. And what happens when we express DUX4 to very low levels and tip them over, no, they're on a pillow. Don't worry, they're soft landing. They're doing fine. But that's a measurable outcome. And this is very low levels. Now, actually, we have a mouse expressing the human DUX4 gene with an assayable phenotype. Without getting into that, it's actually a pretty big deal that a lot of pharmaceutical companies now want to use for testing their therapeutic. Because if that mouse doesn't fall anymore, or he hangs a little bit longer, that's really good news, and that goes for more investment. We did it for CRISPR. That's why I got it. What we showed is single injection of a virus expressing our CRISPR off mechanism, we can actually dial DUX4 down about 30%. Okay, it's not off. Is 30% though enough to get you asymptomatic? Can this keep you out of a wheelchair for 10 more years? That would be pretty good. That's actually our first trial, and we're actually, with the help of the FSH Society, we are funding more trials on this. All right, so basically the take-home message, our understanding of the pathogenic mechanism, the basic biology, understanding how does FSHD lead to pathology, how is DUX4 regulated, have all provided targets. We're using CRISPR-Cas, we're using myostatin inhibition. I say we as a field, this is a worldwide effort. You see me talking, 70 other people can give this talk, right? There's a worldwide effort, a large part because of the FSH Society that provides initial funding for these projects. We have anti-inflammatories, we've got small molecules targeting regulators. It just goes on and on, okay? This is actually really good news. Understanding the mechanism is key for therapeutic development. And so now I think we're kind of leaving the age of what's going on, and we're really entering the age of we can do something about it. Okay, we're going to give Dr. Day's clinic, say hopefully you say the first step one was we need a treatment, right? It's great to have all that. We need the treatment. Well, I think that treatment's not that far off. Okay, I really think, and I think Stephen would agree with me.
I never cared much for moonlit skies. I never winked back at fireflies. But now that the stars are in your eyes, I'm beginning to see the light.
I had to put that in there because he's a good friend. And come to the concert tonight. I hope everybody's going to be at the concert. We're funded by a number of societies. Special thanks to FSH Society. We're a small group. Takiko's made the mouse. And then finally, I answer every email I get from patients. You got a question about the science or what's going on, I may not get back to you right away, but please feel free to contact me. We're happy to answer the question. You can also check out some information on our lab website at UNR. We're very happy to be here on the West Coast now and looking forward to integrating with the UC Davis groups and the Stanford groups now that we're here. So thank you.